Okay, nice meeting you. I appreciate organizers to introduce ToolGen. We are Korea-based biotech company with the development of the multiple genome editing technology, including CRISPR. During last few years, ToolGen has been continuously evolving to apply our CRISPR technology in therapeutics and agricultures. I'm looking forward to share what we have been doing and also where we are heading to for the next few years. Since we are listed in the public stock exchange, so I included this disclaimer in relation to this presentation. ToolGen was founded in 1999 by Dr. Jongmoon Kim, who is now one of the leading scientists in the genome editing field. Currently, we are listed in the KONEX market with a market cap around $150 million US. We managed to secure funding around $6 million US in last couple of years, and with this, we are aiming to translate genome editing platform technology to innovative therapeutics and agricultural product with a vision of improving many aspects of the human lives. For this presentation, we will be focusing on the work in the field of the therapeutic genome editing development. There are two major themes of ToolGen. First, from GFN, TALEN to CRISPR-Cas9, we have been successfully developing important genome editing technologies and are continuously trying to improve current tools. Second, we strive to translate our own genome editing tools to novel therapeutics to cure disease that are untreatable with current approaches. To support these two grand goals of our company, we emphasize two vital factors. We have excellent scientists in our R&D department who have a diverse backgrounds ranging from basic genetics to translational medicine. Furthermore, we filed and will be managing IPs for our platform technologies and their applications. Recent spotlight on genome editing technology. Many of you will be aware of the different tools in this area and their values in biomedical field. Last 15 to 20 years, we have been experienced improvement of a programmable nuclease platform from GFN, TALEN to CRISPR-Cas9, which is enable researchers to freely edit genome with very high efficiency. Because of its simplicity and the reproducibility, we are currently focusing on CRISPR-Cas9. To be more specific, CRISPR-Cas9 involves two key components, a guide RNA which directs Cas9 enzyme to cleave a specific gene sequence. By utilizing CRISPR, we can apply diverse types of genome editing, such as introducing site-specific mutation, specific deletion of genetic element, and correction of a mutation to build therapeutic strategies for various human diseases. These platform technologies of ToolGen is protected by our own IPs. Founded based on zinc finger technology, we have been independently developed three landmark genome editing tools, GFN, TALEN, and CRISPR-Cas9. During this period, we have filed a number of patent applications related to all these genome editing tools. With the rise of the CRISPR-Cas9, our IP strategy has transitioned into the CRISPR-associated genome editing tools. I'm sorry. In year 2012, we managed to edit eukaryotic genome editing using CRISPR. We filed a fundamental patent on this technology, which are currently on the national phase in 10 different countries. Our CRISPR IP has much value because it is based on a very early provisional describing successful human genome editing with this. We had encouraging news from multiple countries of the PTOs. Recently, we had a very good news from Korean PTO that our core CRISPR patent was issued. We believe and are very confident that this is the first of the many good news to follow on securing the value of our CRISPR IP internationally in coming years. Surely we are working on building our IP portfolio for improvement and the application of CRISPR technology for therapeutics field as well. Using CRISPR-Cas9 platform technology, we seek to develop the novel therapeutics. Like others in this field of the gene therapy and genome editing therapy, we are utilizing two therapeutic strategies using CRISPR-Cas9 system for in vivo gene therapy program. We package both sgRNA and our own Cas9 variant into single AAV vector. These therapeutic vectors can be applied either systematically or locally to treat various disorders such as neurological, blood, and aging-related disease. For ex vivo cell therapy programs, we are currently focusing on using CRISPR-RNP complexes that contains sgRNA recombinant Cas9 protein to develop T-cell platform for cancer therapeutics. Indeed, our preliminary studies demonstrate that the electroporation of the CRISPR-RNP complexes resulted in efficient genome editing in human T-cell. We are now consistently achieving between 50 to 90% genome editing efficiency, and the edited cell grow very well after a couple of days of the recovery period. Our initial focuses are developing unchecked T-cell platform for solid cancer by directly and indirectly manipulating checkpoint process. To enable in vivo gene editing, we are using small CRISPR-Cas system that can be fit into one AAV vector, which is very difficult with a conventional S-Biogen Cas9 due to its large size. Our preliminary studies show that our small Cas9 system can support efficient and specific gene editing in cultured cell and also in animal. This efficient single vector system can be applied to a diverse range of diseases of specific organ, such as the liver, eye, and nervous system. Through our initial in vivo therapeutic programs, we are anticipating establishing preclinical efficiency in animal model by the first half of next year. The above-mentioned ex vivo and in vivo genome editing technologies can be applied to a various range of diseases. However, due to the limitation of the time and resources, we are focused on few therapeutic programs. We are performing and also plan to perform the preclinical test of our early stage therapeutic programs in animal models over the next year or so to establish in vivo efficacy and examine potential off-target issues. And once high efficiency and low toxicity profiles of these therapeutic programs are established, we will either undertake the clinical trial ourselves or seek licensing opportunity with others. We do not do these programs alone. Therefore, we continue to seek for strategic partners to hasten therapy development. We strongly believe that the collaboration with the scientific experts are essential and value-enhancing. Currently, we are working with a renowned neurologist at Samsung Medical Center to develop therapeutics for peripheral neuropathy, and geneticists at the Seoul National University to treat the hemophilia A and liver-associated disease. And we are also working with Green Cross Cell, which is a company who expertise in T-cell-based cancer therapeutics. Other than above-mentioned therapeutic platforms, we also have some more programs in earlier stage of development for which we are looking for collaboration partners to make research more economical and to quicken the pace of the delivering potential product into clinic. Based on business partnering is of utmost importance to us, so we are open to any company that would like to work with us to bring our programs or also new exciting programs into clinic and eventually into market. Our future plans include the development of the better or next generation tools for genome editing, making proof of principle studies of our therapeutic programs available, and preparing IND, and also establishing strategic partnering to expedite the process and maximize the value of the therapeutic genome editing development. And this is my end of presentation, and thank you for listening. And please contact me if you have any question about our technology and R&D and our business plan for partnering. Thank you.